Hello friends, and in this video I will be discussing a top 25 topics in general microbiology which have been asked in examination, and this video is exclusively for MRB candidates. So, and have highlighted the important points, okay, so which have been highlighted like this. See, I have put in blue color, and this have been asked in the previous year examinations. So the first, as you know, always they will be asking some topics in the history of microbiology. In that, the first is father of microbiology is Louis Pasteur. So again, friends, I want to say I have posted a separate video on this scientist who contributed in microbiology, and I'll post the link below. Okay, so I don't want to waste the time. So the first is a father of microbiology is Louis Pasteur. So father of microscopy is Anton von Leeuwenhoek, and Louis Pasteur, there is another point, and he proposed the method of sterilization and the fermentation. Okay, so the father of bacteriology is Robert Koch, and father of chemotherapy is Ehrlich. You can remember like so. Next, and another is he had discovered a bacteria called Ehrlichia. Okay, so he is called as a father of chemotherapy, and here sepsis, that is the father of aseptic surgery is Joseph Lister. You can remember like, see here SC, here also SC. Okay, so it is a Joseph Lister, and electron microscope it's EE. Okay, Ruska, and smallpox virus it is Edward Jenner, and PCR by Mullis. Okay, so phagocytosis is by Mechnikov, and air forms of bacteria it is mainly in the Kleinberger, and transposons is by Barbara McClintock, and DNA sequencing is by Gilbert and Sanger. So friends, you must not miss any of the scientists over here. Anything can be asked in exam, but these are the most commonly it can be asked. Okay, so second point is type of light which is used in microscope. So in bright field microscope we will be using a transmitted light, in dark field we will be using a reflected light, and in interference contrast microscope we will be using a polarized light, and in electrons will be used in the electron microscope. Okay, so they will be asking like what kind of light can be used in bright field. So they will give the options, and here the correct answer is a transmitted light. Okay, so third point is about capsulated bacteria. So here it is a pneumococcus, meningococcus, Haemophilus influenza, Klebsiella pneumonia, and Pseudomonas, and Bacillus anthracis. So here the one point you want to remember here is the capsule of all the bacteria is usually made up of polysaccharide, except it is in Bacillus anthracis, it is made up of a polypeptide. So that have been asked in exam. Okay, so I have highlighted the previous year questions. So next here, capsulated fungus is Cryptococcus neoformans. So another point you want to remember here, the Histoplasma capsulatum. So the name is having a capsulatum, but it is a misnomer. It is not a capsulated organism. Okay. So next, coming for arrangement of flagella. So here, so the monotrichus is nothing but the bacteria will be having a single polar flagellum. Okay, only one flagellum will be there, and here the example is, I think you will know polyvinyl chloride. So P for Pseudomonas, V for Vibrio cholerae, and C for Campylobacter. So next, Lophotrichus, it is a spirillum. Just remember LS. Okay, lady security or whatever it can be. So Peritrichus flagellum, they will be having a flagella over the entire body surface. The example here is E. coli and Salmonella typhi. And for Amphitrichus, there will be a single flagellum will be there at the both ends. If you remember, it is a gram negative bacillus. Okay, they will be having a single flagellum at both the ends. Okay, so coming for next, it is an So again here they have asked the, sorry, I forgot to highlight this. So the Vibrio cholerae have been asked in the examination. So next point is the types of motility. So here always remember a tumbling motility is present in the listeria and gliding motility by mycoplasma and stately motility by Clostridium, darting motility by Vibrio and Campylobacter, and swarming by Proteus and Clostridium tetani, and corkscrew lashing and flexion extension by spirochetes. So friends, again I want to say one point here, that is, so here they can either give tumbling motility or the differential motility. It can, whatever it can be given. Okay, so because listeria will be showing both that is a tumbling and the differential motility. So you want to remember the both the terms. Okay. Sorry friend. So just remember these two terms. Okay, thank you. So next it is a generation time. So what is a generation time is nothing but, so it is a it's a time required for the bacteria to give rise to two daughter cells. That is in generation time. So always remember for E. coli it is at 20 minutes, M. mycobacterium tuberculosis it is in 14 hours, and for lepre it is in 13 days. Okay, so always remember the lepre it will be having a longest incubation period. And just you want to know this, and they have asked for Escherichia coli. Okay. So next it is in bacterial growth curve. It is an important topic. So here, so first always remember there is a 4 phases. So one, it is an lag phase, log phase, stationary phase and decline phase. So lag phase, it is nothing but, just remember the cells will be attaining a maximum size and there will be an increased number of the enzymes and metabolites. Whereas in log phase, all the cells will be uniformly strained and it will be active stage. Whereas in stationary stage, it usually produces all the granules, spores, exotoxin, antibiotic and the bacteriocin. So here decline phase, it usually produces an involution form, that is an aberrant forms. Okay, this usually takes place in the stationary phase, that is an S for S, you can remember. So spores it is an S and again the stationary it is an S. Okay. So coming for next topic is oxygen. So here, so all the organism will be requiring oxygen for the basic requirement, but there will be in some of the exceptions. Okay, so we'll be seeing that. So first it is an obligate aerobe, means this organism usually needs an oxygen. They can't live without an oxygen. Example, it is in Pseudomonas, tuberculosis, Bacillus, Brucella and Nocardia. So again coming for facultative anaerobes, you can remember like this is if they have given anaerobe. Okay, so facultative term, if they have given anaerobe, so just remember they can grow anaerobically, but they are actually aerobes. If they are given facultative aerobe, they can grow aerobically, but they are actually anaerobes. Okay, so for facultative anaerobes, example is E. coli and Staph aureus, and for facultative aerobe, it is lactobacillus. So coming for next topic, it is a microaerophilic. They can grow only in the little amount of oxygen, that is in 5 to 10%. It is usually, you can remember like MHC. Again here M, here also M. MHC is Mycobacterium bovis, Helicobacter and Campylobacter. So coming for obligate anaerobe, they do not require an oxygen. If there is an oxygen, it is lethal for them. Okay, you can remember like that. So here it is in Clostridium. So next point here, you want to remember this. The aerotolerant is, they can tolerate some amount of an oxygen, that is in Clostridium histolyticum. Okay, so now we have discussed, in this you want to know the definition and the examples for each type. And next it is about the temperature. The temperature is divided into 3 types. One is psychrophiles, mesophiles and thermophiles. So coming for psychrophiles, So group first it is it will be growing under 20. If it is 20 degrees Celsius psychrophile, meso is 25 to 40 and thermophile is 55 to 80 degree. Here the example is most of the pathogenic bacteria, and here it is in Bacillus stereothermophilus, and this have been asked in exams. Okay, so what's an example for the thermophilic bacteria? So next topic is what are the some of the give the examples for the obligate intracellular organism. So here the first is a bacteria like Mycobacterium leprae, Rickettsia, Chlamydia and Coxiella, and all the viruses are usually obligate intracellular. The fungus, it is a Pneumocystitis jirovecii, and a parasite, it is a Toxoplasma, Cryptosporidium, Plasmodium, Leishmania, Babesia and Trypanosoma. So you can remember like, see, Leishmania and Trypanosoma is I will be reading the two, and Toxo and Cryptosporidium will be coming under one topic, and Plasmodium and Babesia. If you see the parasitology book, it will be coming in this order, so you can remember like this. Okay, if you're remembering Leishmania, then obviously Trypanosoma. So coming for the next topic, it is a sterilization and disinfection, and this have been asked in exams. So here the first you want to know the definition. So sterilization is nothing but it usually destroys all the microbes. S for yes, you can remember, that is it can destroy all the microbes including the spores. Yes for yes, sterilization and spores. So coming for disinfection, it usually destroys only the microbes and not the spores. Coming for asepsis, so here the agent will be, some of the chemical agent will be applied to the body surface, and decontamination makes it stiff. So it is mainly for the some of the items to take it very safe and to handle. Okay, so just remember these two definitions, that is in sterilization, disinfection. It's the most common. So now, so actual classification is divided into one physical method and chemical method. Okay, so coming for physical method, so first it is in heat. So under heat, you want to remember dry heat and moist heat. So dry heat, it is in flaming, incineration and hot air oven, and for moist heat it is temperature less than 100, at 100 and more than 100. So less than 100 is pasteurization, water bath and inspissation, and for what is that, at 100 is boiling, steaming and tintalization, and more than 100 is autoclave. So friends, again I am saying this anything can be asked. Okay, so here each and every point is important in this classification. And for filtration, it is a depth filter and membrane filter, and for radiation it is an ionizing and non-ionizing. So ionizing, it has X-rays, gamma rays and cosmic rays. So non-ionizing has UV and IR rays. Okay, so next it is an ultrasonic vibration. So here you want to remember everything. And coming for chemical method, so first it is an alcohol, ethyl alcohol and isopropyl alcohol, which have been used in the sanitizers. And coming for all aldehyde, formaldehyde, glutaraldehyde and orthophthaldehyde. The formaldehyde has been used in the fumigations of OT, and glutaraldehyde, that is in 2%, it is called as in Sidax. It is used for the endoscopes and all. Again, this have been asked in examination. So phenolic compound of Trazol, Lysol, chlorhexidine, chloroxylenol and hexachloroprophene. And halogens are chlorine, iodine and iodophore, and oxidizing agents are hydrogen peroxide and peracetic acid. So coming for salt, it is a mercury chloride and copper salts. Sorry friends. Okay. So surface active agents are quaternary ammonium compound and the soap. And coming for dyes, it is an aniline dyes and the acridine dyes. Gas sterilization, it is a low temperature steam formaldehyde and ethylene oxide and beta propyl lactone, that is called as in BPL. Okay, so here, so the important points you have to remember here is, you want to by heart the entire classification, and some of the important point is what is the mechanism of the each sterilization. That is, for dry heat you can remember like code, that is charring, oxidative damage, denaturation and elevated level of electrolytes. So coming for the moist sterilization, it is only by denaturation and the coagulation of the proteins. Okay, so you want to remember these 2 points and okay. So coming for next topic is that is in pasteurization. Okay, so I'm not explaining all the classification, so I'm explaining only the point which is an important. Okay, so coming for pasteurization, as you know, this term is only for the milk and the dairy product, and here there is a 2 method. One is a Holder method and another is in flash method. So Holder, it is in 63 degrees Celsius for 30 minutes. This have been asked in exam. And flash method, it's 72 degrees Celsius for 20 seconds and followed by cooling to 13 degrees Celsius. So here this is an important. Okay, so again, so all non-sporing pathogens are killed except Coxiella burnetii, and again this have been asked in exam. Okay, so you want to remember the temperature for Holder method. So you can remember like Holder means it is an when we'll be holding is like usually at the older ages, right? So you need some of the person's helps to walk, or you'll be using less stick. So the old age most commonly is about 60 to 63. So you can remember like that. Okay. So next is some of the important temperature you want to remember here. One is an hot air oven, it is a 160 degrees Celsius for 2 hours, and for autoclave it is in 121 degrees Celsius for 15 minutes at pressure of about 15. Okay, so you want to remember these 2 temperatures. Okay, so friends, I'm saying you must not miss a single point from this video. Okay, so I have selected only the important points only. So coming for testing for disinfectant, there are 4 tests. One is phenol coefficient test, another it is a Chick-Martin test, and there is a Kelsey-Sykes test, and there is an in-use test. Okay, so that's just a modification of the Kelsey-Sykes. You don't want to details of the test, just remember the names. That is more than enough. Okay, so next topic, it is in culture media. First it is in selective media. Okay, so solid media, what is a selective media is, so it will be allowing only the pathogens, not the normal commensals. Okay, it's a solid media that allow only the pathogen and it will inhibit the normal flora. So what are the examples here is, one is Lowenstein-Jensen for Mycobacterium tuberculosis, and thiosulfate citrate bile salt sucrose for Vibrio species, and for Shigella, Salmonella, it is an deoxycholate agar and XLD. Okay, so potassium tellurate, you know, as it is for the Corynebacterium diphtheriae, and Wilson Blair medium is for the Salmonella typhi. Okay, so this in this LJ medium have been asked, friends. Okay, so another important point about LJ medium I will discuss at the end. So coming for differential media, it is in McConkie agar and Clyde agar. So what is differential medium is, you can differentiate between the lactose fermenting and non-lactose fermenting colonies. So whereas lactose fermenting colonies will be producing in pink color, whereas non-lactose will be producing in colorless colonies. Okay, so coming for enriched media, so first it is in, what is an enriched media? So to the basal medium we will be adding some of the nutrients, like it can be egg, it can be blood, or it can be serum, to make it supportive for the growth of the bacteria. Okay, so that is a definition. So example here are the blood agar, chocolate agar, and Loeffler's serum. Okay, so here you want to remember, so blood agar you will be using in sheep blood agar. The percentage is about 5 to 10%. The temperature is at 45 degrees Celsius. So here you can demonstrate the hemolytic property of the bacteria, that is alpha hemolytic, beta hemolytic, and gamma hemolytic. So coming for chocolate agar, it is nothing but it is a heated blood agar, and here the temperature is about 70 degrees Celsius, and it usually supports a Haemophilus influenzae. So Loeffler's serum, you know, it is mainly for the Corynebacterium diphtheriae and potassium tellurate agar. As I mentioned, so coming for enrichment broth, so here it is again same like an enriched medium, but it will be in a liquid form. Okay, so that is a broth. So here the selenite and tetrathionate broth for Salmonella, Shigella, and alkaline peptone water, it is mainly for the Vibrio. So coming for transport medium, so transport medium is mainly for the transport of the organism, so without causing the multiplication. Okay, so first it is in Pike's medium for Streptococcus, Ames and Stott for Neisseria, Vibrio cholerae for Venkatraman Ramakrishna medium for Vibrio cholerae, Carrie Blair and autoclaved seawater, and buffered glycerol saline and Carrie Blair medium for Shigella and Salmonella. So here next it is in. So coming for PCR, as we discussed already, it was discovered by Carrie B. Mullis. So what are the 3 steps is extraction, amplification and gel electrophoresis, right? So here the amplification has 3 steps, that is an important here: denaturation, primary annealing and extension of the polymer. Okay, so denaturation is nothing but the separation of the DNA, and so here there will be a temperature is 95 degrees Celsius, annealing is 55 degrees Celsius and extension of primer is 72 degrees Celsius. Friends, you want to remember what are the steps and what are the steps involved in amplification and the temperature. So coming for next topic is bacteriophage typing is done for 5 bacteria. So one, it is an Staphylococcus aureus, Salmonella typhi, Vibrio brucella and Corynebacterium diphtheria. So next, so gram-positive silver. So always remember it will be having a thick peptidoglycan, whereas gram-negative will be having thin peptidoglycan. Here there is an absent lipopolysaccharide, aromatic amino acids and lipids. But in gram-negative lipopolysaccharide, it is a main component and it is an important component for gram-negative bacteria. And teichoic acid is present in the gram-positive, whereas it is absent in gram-negative. So remember here, LPS is usually present in the gram-negative bacteria, whereas teichoic acid is usually present in the gram-positive bacteria. So coming for intrinsic antimicrobial resistance. So what is that? Is nothing but it is an innate ability. That is naturally the bacteria is resistant to some of the antibiotics. Okay, so it is an innate ability of an organism that will be exhibiting a resistance towards a certain species, certain antibiotics. So first here is the anaerobic bacteria. It is generally shows an intrinsic resistance to aminoglycosides, and aerobic bacteria is mainly for metronidazole. Gram-negative bacteria are usually resistant to vancomycin because vancomycin it is used for gram-positive bacteria. So Klebsiella species is for ampicillin, Pseudomonas for sulfonamides, trimethoprim, tetracycline and chloramphenicol. So here this have been asked, okay, metronidazole. And enterococci have been resistant for aminoglycosides and the cephalosporins. So these are all the compilation of the intrinsic resistance. So next topic here is what are the antimicrobial susceptibility testing methods. So one, first it is in this diffusion method. We will be using in Kirby-Boyer method. Okay. Next it is in dilution test, that is a micro dilution and macro dilution, and epsilon meter test. What are the automated is PCRs? Automated is Backtech method, Whitetech. Molecular it is in PCR. Okay, so just remember what are the methods have been used for the antimicrobial susceptibility testing. So again, this is an important topic. So the southern blotting is for DNA, northern blotting is for detection of RNA, western blotting is for protein. So friends, remember like it is a snowdrop. S-N-O, you can't remember, W, here D-R-O-P. I think you remember this famous mnemonic. Okay. So another there is an important, that is an eastern blotting. Okay. So the eastern blotting is for the lipids. Okay. Okay. So next. So friends, as we discuss the methods of sterilization above, so I'll be saying some of the important methods which have been used and asked in examination. So as you know, milk is usually for sterilization, it is by the pasteurization. So ionizing radiation is for plastic syringe, catgut suture, swab, catheter, bone and tissue graft and adhesive dressing. So this have been asked. Okay. So next for culture media, metal instrument, glass glass and suture material except catgut is autoclave, whereas catgut it is by the ionizing radiation. So paraffin glass. So again friends, I want to say the culture media all are usually sterilized by autoclave except LJ medium, that is a Lowenstein-Jensen medium, it is done by the insufficiency. Okay, so as before I told you, I will be saying an important point about LJ medium, that is one point. So paraffin, glass syringe and flask slide oil, grease, fat dissolved by hot air oven. So friends, again this. So the hot lung machine, respirator, dental equipments are sterilized by ethylene oxide, and heart lung machine have been asked repeatedly. Okay. So next, coming for bacterial genetic. So friends, here you want to remember three terms. One is transformation, transduction, lysogenic conversion, and it is a conjugation. Okay, so first it is a transformation. What happens here is the transformation of genetic material is through free DNA. So next it is a transduction. Here the portion of DNA, so that is a transfer, is usually by the bacteriophages. Okay, so the penicillin resistance in Staphylococcus, as you know, that is an, what is a penicillin binding protein? MEC to MEC-A, it is usually mediated by this process, that is a transduction. What is a lysogenic conversion is nothing but, so here what a transduction will be, the bacteriophage will be helping, but here mainly the phage DNA, that itself will be confers the genetic information to the another bacteria. Okay, so transformation is free DNA, transduction is by bacteriophage, what the lysogenic is by phage DNA, and conjugation, you know, as it is done by it's a mating of the male and the female, it is usually by the conjugation tube, and this it is an important for drug resistance. Okay, so again friends, this have been asked in exam, the highlighted. Okay, so we want to know the definition for all. So next, so that's a famous topic. What is the difference between exotoxin and endotoxin? So here just remember one, either exo or endo. Okay, if you remember exo, it is a protein, so opposite is lipopolysaccharide. They will be diffusing into surrounding medium, so they do not diffuse. Here it has an enzymatic action, there is no enzymatic action. Highly antigenic, weakly antigenic. It is not a fever inducing, that is not pyrogenic, here it is a pyrogenic. So here all the gram positive bacteria will be producing an exotoxin. The exceptions are some gram negative like Shigella dysentery, Vibrio and etc. That is an toxigenic E. coli. So here endotoxin is produced usually by gram negative except Listeria, that is a gram negative positive bacilli, right? So it will be producing an endotoxin. So exotoxin is produced by gram positive organism, but exception is Shigella dysentery, Vibrio cholerae and etc. Whereas endotoxin is mainly produced by gram negative organism, but exception is Listeria, that is a gram positive bacilli will be producing an endotoxin. So coming for the more again an important topic, that is on biological indicator. So if you are doing sterilization, we want to know whether it is an valid or what is an accuracy. So for that we will be using some of the indicators. So again friends, you want to remember, I can't help you. So first it is an hot air oven, it is a Clostridium tetani, toxigenic strains, that is non-toxigenic strains, and Bacillus subtilis subspecies niger. So but for here autoclave have a mnemonic, so the auto will be having a steering, right? So Geobacillus stereothermophilus. So filtration, it is a Brevantimonas diminuta and Serraceae, and ionizing radiation, it is in Cumulus. So friends, you can remember, I'll think see iron, so right, so Pume is nothing but you can remember like Bhumi. So it is an Bhumi is nothing but it is an earth in Tamil. So we will be getting an iron from the earth, you can remember like that. Ethylene oxide, it is an Bacillus globigi, and plasma sterilization Geobacillus stereothermophilus, and again it is a subspecies niger. Okay, so friends, you want to remember these points. So coming for plasma sterilization, so here it is an okay. So here first it is a plasma sterilization. You want to remember what is the agent we will be using is hydrogen peroxide and peracetic acid. So it is mainly for the heat labile, and that is a surgical instrument, as low temperature is usually maintained at 50 degrees Celsius. It is not as much high like hot air oven or the autoclave. And here the sterilization control is Stereothermophilus and the Bacillus subtilis subspecies niger. So friends, and so I'm again repeating, please don't miss a single point from this video. And second point is, I did not cover this training procedure because I have separate video for this training, and I will be giving a description box below, and for scientists also I will be giving a description box below. So if you go through these points, I'm damn sure some questions will be asked from the general microbiology from these topics. So friends, please do not miss any single point from this video. Okay.